Welcome to our video presentation on auxin, done by Matt, Eugene, and Alex. Auxins are plant hormones found in all parts of the plant. They are the first of the major hormones that have been discovered. The term auxin derives from the Latin word auxin, and it means to grow or increase. The functionality of this compound is greatly determined by its concentration in each position. It participates in major processes such as cell elongation, wall relaxation, cell differentiation, ethylene production, inhibition of root growth, and flowering. There are various types of naturally occurring auxins. Chemically, they are all composed of an aromatic ring and a carboxylic acid. However, it has been found that aldyl-3 acetic acid is the most important in the growth and development of plants. So we will focus mostly on this variation. In recent times, scientists have created synthetic variations of auxin, including IBA, NAA, and 2,4-D. These are used in agriculture for promotion of root and fruit growth, but mostly for controlling broadleaf weeds in grass crops and pastures. The discovery of auxin started in 1881 by Charles Darwin and his son Francis. They performed a series of experiments on coleoptils. This is a protective shelf that covers emerging shoots in grass type of plants. They exposed the coleoptils to light from an unidirectional source and observed that they bended towards the light. When the tips of the shoots were removed, they observed that the coleoptils did not grow towards the light source. When the tips were covered with an opaque cap, the shoots did not bend towards the light either. Darwin concluded that the tip was responsible for sensing light and that a messenger is transmitted in a downwards direction from the tip of the coleoptile, causing it to bend. In 1913, Danish scientist Peter Boysen Jensen demonstrated that the messenger was not transfixed, but mobile. As Darwin did, he cut the tips of the coleoptiles and observed that there was no bending towards the light. Next, he inserted a gelatin between the tip and the shoot that prevented cellular contact but allowed chemicals to pass through it. When he did this, he observed that there was positive bending towards the source of light, meaning that the messenger was mobile. In 1926, Danish botanist Fritz Wendt defined the messenger substance as a growth-promoting hormone. He performed an experiment where coleoptile tips were placed on agar with the objective of trapping their chemicals. The agar was then cut into blocks and placed onto the decapitated stem. One group had blocks evenly placed, and the other had the blocks placed unevenly on one side of the coleoptile only. Fritz found that evenly distributed messengers promoted straight growth. However, when the messengers were unevenly distributed, he observed that the shoot curved away from the side with the hormone. He then realized that this substance promoted growth and decided to name it auxin. Without knowing it, these researchers would describe the first mechanisms of what is now known as phototropism. Modern biologists have understood that most plants have the capacity to adapt to their environments by growing towards the light source and obtain the optimal position for growth. It is now known that auxin is the main conductor of this process, thanks to its cell elongating properties. When sunlight is overhead, the auxin molecules are distributed evenly and the shoot grows straight. Once the sunlight shines at an angle, the auxin molecules move to the far side and induce elongation of cells on that side. Then the elongation of the cells on one side results in the bending of the shoot towards the light. This mechanism is extremely important for plant survival. Most plants grow in competitive environments and they have to fight for the availability of light. In this next segment, we will show you a time-lapse of young tomato plants that are following the light throughout the day in order to obtain their best position for light absorption. will talk about the cell elongation properties of auxin. It has been found that the acid growth system is responsible for this elongation process, and it is done by altering the plant wall plasticity. Inside the plant cells, plasma membranes contain protein pumps responsible for the flow of hydrogen protons towards the cell wall. Auxin hormones stimulate these hydrogen proton pumps, and consequently the secretion of hydrogen ions decreases the pH levels in the cell wall. The acidified cell wall activates pH-dependent enzymes and breaks bonds between cellulose microfibrils. This causes the wall to loosen and the water pressure to expand the cells. Another auxin-induced process that is essential for survival is apical dominance. It refers to the growth dominance of the central stem over the side limbs. Auxins located in the shoot apex inhibit the growth of lateral buds so that the plant may grow vertically. This process starts in the early stages of growth, and it forces the plant to use available energy for growing upwards. And this is very important for maximizing light exposure. In this diagram, we can see the presence of IAA hormones in the apical bud. They maintain the stored carbohydrates in the meristem. Once the apical bud is cut off and IAA molecules are not present, the sugars get evenly distributed to the buds, and new limbs grow. This is the reason why botanists strategically cut off the apex in plants. It promotes more branching, and this allows for more flower, leaf, and fruit formation. This picture shows the lateral buds being deprived of energy. In the right picture, the apical bud is removed and the lateral branches are much more developed. In the next time-lapse video, we will see normal apical dominance. At this stage, the apical bud is removed and auxin hormones stop inhibiting growth of lateral limbs, so we can slowly see the formation of new shoots. Now we'll briefly talk about the modern uses of synthetic auxin. Scientists have developed various forms of synthetic auxin that are used in modern agriculture. Some of these products are used as herbicides that are effective at suppressing difficult to manage broadleaves, and others are good at providing brush control and also controlling woody plants in forests and non-crop plant plants. 2,4-dichlorophenoxyacetic acid is the most commonly used form of synthetic auxin. This molecule is a selective herbicide, affecting dicot plants but not grasses. This means that when applied to cereal crops, it kills opportunistic weeds without damaging the crop itself. The process by which auxin kills the affected plant is still relatively unknown. However, the current theory is that this hormone forces broadleaf plants into producing excess ethylene, which leads to the flooding and rapid aging of the plants. And this concludes our presentation on auxin. Thank you very much for watching.